Assalamualaikum dear students. Today we will be discussing the process of erythropoiesis. The basic stem cell that causes the production of erythropoiesis is the hematopoietic stem cell, which divides into two progenies. One is the lymphoid progeny, that is the lymphoid stem cell, which further give away the agranulocytes, and secondarily the myeloid stem cells, the ones that will give rise to the RBCs. Now you must be knowing that the myeloid stem cells, they give away three progenies. One is the RBC cell type, the other one is the WBC cell type, and last the platelet cells. Now the myeloid cells giving away or forming an RBC depends on the very important factor that is released by the kidneys due to hypoxia, known as erythropoietin. So erythropoietin is the one that will decide that the myeloid cell will be giving away a pro-erythroblast, that is, it will form later on an RBC. So starting from the pro-erythroblast, and this erythropoietin also will affect all the subsequent steps arising from the pro-erythroblast as well. Now the pro-erythroblast, as we all know, due to the hyperactivity of the nucleus in the pro-erythroblast, there will be massive formation of ribosomes and proteins, hence giving it a bluish color. So the formed cell is known as basophilic erythroblast, that is base loving, which is blue in color. Then as the transcription and the process of translation goes on, there will be now formation of the protein, that is the hemoglobin. So at the polychromatic level, the hemoglobin will appear, so that it's known as polychromatic as it has both the colors, that is, it dyes blue as well as pink. So a polychromatic erythroblast will be formed. Now this polychromatic erythroblast will, as proceed in the process of erythropoiesis, the transcription will stop occurring and the nucleus will condense and mature more, giving rise to a more reddish colored cell that is known as orthochromatic erythroblast. So this cell is known as polychromatic erythroblast. This is the one which has massive amount of hemoglobin present in it. Now this orthochromatic erythroblast will lose its nucleus along with the organelles like the mitochondria. The mitochondria is lost by the RBC so that it may not utilize the oxygen that it transports, and hence a new structure will be formed by the exclusion of the nucleus and the organelles, resulting in the formation of reticulocyte. Now the reticulocyte will remain within the bone for 1 to 2 days and ultimately will enter into the blood stream. This is a blood vessel, and the reticulocyte will then later enter into the blood stream. In the blood stream, the reticulocyte will further go into maturation and a biconcave ultimate disc-shaped RBC, a mature RBC, will be formed. Now let's see how different factors are affecting erythropoiesis. As we have discussed earlier in the previous lecture, that erythropoiesis is affected by the substance iron, vitamin B12, we have folic acid, nutrients, and erythropoietin. Erythropoietin we have I've told you before. Now let's see how does and where does iron act. As iron is a component of heme part of the hemoglobin, so it will be affecting the areas that are producing hemoglobin. Vitamin B12 and folic acid are acting as the substance that mature the DNA and the nucleus, so they are acting at all those levels that are having DNA that are having nucleus to it, while the nutrients are also important in the formation of heme, so they are actively participating in the areas where hemoglobin production is taking place. Now, as we all know that RBCs are not the actual type of living cells in our body, as they lack the nucleus and they lack almost all of the organelles, so they have a very short lifespan, almost of 120 days. As the time passes, the cell membrane of the RBCs, they lose their elasticity and they lose their biconcave shape, and they become more of a rigid, star-like or a spherical sort of a cell. Now, due to this change in their shape, they can no longer squeeze through the blood vessels, especially of the spleen, the bones, and the liver, where we have these typical reticuloendothelial cells and sinusoidal vessels, that is the capillaries that have large pores through them. Whenever the RBCs, these aged RBCs are trying to pass through these sinusoidal spaces, they get stuck over there, and the macrophages that are part of the reticuloendothelial system of these organelles, they engulf the RBCs and they destroy the RBCs. Whenever these RBCs are destroyed, then the hemoglobin is degraded into its initial forms, that is the heme part and the globin part. The globin is an amino acid and is recycled, while the heme part gives away the iron along with the bilirubin. Iron is again recycled, while the bilirubin, which is an orange colored substance, enters into the liver. This bilirubin is a non-conjugated or an unconjugated bilirubin, which when reaches to the liver, it combines with a glucose containing substance which is known as glucuronic. So bilirubin combines with glucuronic acid, gives away the conjugated bilirubin. Now this conjugated bilirubin then enters the duodenum through the bile duct, and in the duodenum, the bacterial flora of the duodenum causes the degradation of bilirubin and converts it into a new substance which is known as urobilinogen, and then this urobilinogen is excreted out of the body through the feces or through the kidneys. So that was it for today. I hope so you have understood.